Hello and welcome to this Aston Originals podcast. It's been a little while since you've heard from us at the Aston Institute for Membrane Excellence, but we are back, don't worry. Hope you've not missed us too much. And today's instalment, we have some wonderful people with us. I'll first introduce myself, not as a wonderful person, but you can make your own mind up. I'm Dr Matt Derry. I'm a senior lecturer in chemistry here at Aston, and in the Aston Institute for Membrane Excellence, I'm a member. And with us, we have Dr. Emma Shepherd, who's also an associate member of AIM and a lecturer in biosciences. We've got Dr. David Jenkins, who's an AIM research fellow. And we've got Dr. Dominic Mosses, who's a postdoctoral research associate in the College of Health and Life Sciences. Welcome. First podcast for all of you.
Yeah, thank you.
And today we're here to discuss so AIM spans from chemistry all the way through biochemistry up to molecular biology and cell biology. And we're more towards the molecular biology, cell biology type today, and talking about a relatively new instrument and the only one of its kind in the UK, which is the Quantum X high resolution 2-photon 3D bioprinter, which is a mouthful. So we're going to break that down for you and hopefully explain exactly what that is. This instrument was made possible by BBSRC, so that's the Biological Sciences Research Council Alert 2022 bid, which was led by the legend that is Professor Rain Parry. And Emma here was a co-investigator on that grant as well, so can give us some information on that. And here is the instrument itself, and that's what just over £600,000 gets you. But inside there's a lot of fancy tech, which we're going to break down for you. So I think the obvious place to start, and I'm going to ask you, Dom, is what on earth is a high-resolution 3D bioprinter?
Yeah, it's a mouthful. So bioprinting in itself is everyone's sort of familiar with 3D printing. You're making a 3D sculpture with the material. Bioprinting is the ability to incorporate cellular material, organic matter, and elements of our body into the print. If you think about this printer, it's like a nano sculptor. It's a very tiny sort of way of sculpting a structure with cells inside, and with it we can recapture many elements of the body that we can't do with sort of other 3D printing processes. Bioprinting spans a variety of different sort of 3D printing techniques. There's a lot out there, but this one allows us to have a very, very high degree of control and sort of precisely position cellular material in the areas that we're printing. It's a bit different to sort of anything that's out there at the minute. Yeah.
Okay, cool. So the phrase 2-photon printing was mentioned as the thing that I guess makes this happen. And I know, Emma, you've got a cool analogy that explains what on earth 2-photon printing is.
As Dom says, it's a fancy advanced 3D printing technique. And what we do is use laser light to cross-link to drive this polymerization that causes a tiny little structure that we can control. So if we think about how these things print, we have photons coming from lasers that only will conduct this polymerization reaction if 2 photons hit a molecule at exactly the same time. And the reason why this works is we use a material that will only polymerize in those conditions. And so if there's just a single polymer floating through, no reaction will take place. And it's only this specific 2 photons hitting at the same time that causes this very restricted polymerization. And so if you want my Star Wars jam analogy,
please.
Here it comes. I'm sure you don't, but I'll just plow on anyway. If we think I'm going to make some Star Wars Jedi jam, and I've got my starting material is my Jedi jelly, and so it's like a clear, sticky, kind of syrupy material. And traditional ways that we know is how I'm going to polymerize this is I'm going to use a lightsaber. And this gel will only form a jam if I've got my lightsaber there. And so if we think about some of the traditional printing methods, we'd go in with quite a severe sort of my biggest, strongest lightsaber. And everywhere that goes into that gel will form a polymer, and that will form a gel. So that will make the jam throughout. But what this technology does is imagine we go in with 2 really weak lightsabers. I can go through that gel within each individual lightsaber, nothing happens. Only at the point where the 2 lightsabers cross is the energy sufficient for that unique point of polymerization. And so we can imagine me with my 2 lightsabers almost drawing little polymers as making my shapes. And that's really exciting, because that means that instead of just, for somebody like us who works with cells, instead of just going in and imagine back to my crazy lightsaber, we've got a gel and I want to put my cells in there and I come in with my super strength lightsaber, that gel is completely set, all my cells are scorched and burned. But what we can do with this technology is we can etch little shapes, we can make little pockets for the cells to go into, and little channels that we can do lots of cool science with.
Amazing. So let's put into context, what color are your lightsabers?
Purple and green.
Sounds pretty cool to me. And what's also pretty cool is there's a lot of mention of polymers here. And as a polymer chemist, obviously that strikes a chord with me. Yeah, you've got me hooked.
Indeed.
But I guess the real applications is more on the biological side. So what is the instrument useful for, Dave?
So we can use it for different tissue engineering applications. So we can grow cells into specific shapes, whether that be on a 2D surface or in a 3D surface. The world of biology is trying to move towards 3D because it better recapitulates what we see in the body. So most of our cultures at the moment are just done on a 2D really hard surface, which is just not where the body is. So we need to try and make things in the sort of shape that they are in the body and in the 3D structure that they are in the body. So then we can look at how cells develop, and my research focuses around the brain. So I go neurons in this type of thing, and we can get them to connect in ways that we see in the body that we can't get in a normal way. And Emma works on things like the liver to get them in the sort of ways that the blood flows throughout the liver.
Okay, so like more realistic models,
100%.
Yeah. So what? Because another thing that is in that long phrase that I said, what the Quantum X does is high resolution. So what kind of resolution are we talking about?
So the smallest that the company says that we're testing towards getting to is around 200 nanometers in the X and Y. And then I think it's about 1 micron in the Z, something like that. So we can get down to really fine structures that are much smaller than a cell. So we can see what these nanostructures have, the effect these nanostructures have on cells, both if they're big enough to sit inside them or if they're just small enough that the cells can feel them, because they might have different reasons for things to bind to them, whether that be cells or proteins that we put on.
Yeah, so this is that tiny nanosculpture that you talked about. So really fine control of the surface. So this is all about printing around or embedding cells in these structures. So Dom, you're the one that's kind of looking after the instrument. How easy is that?
It's quite difficult traditionally in other 3D sort of print processes, because people have been 3D printing and embedding cells in 3D prints for quite a long time. The issue that you find is when you're taking a jelly to a jam, you sort of lose the degree of control of how much it solidifies. And if it gets too solid, if it gets sort of away from jam almost towards a slab of jammy concrete, the cells don't like it at all. It sort of squashes them, as you can imagine. If you put a human in a tight space, it can be quite claustrophobic, and the cells are exactly the same. And also the processes have been toxic down the years, but with this process, with the process of the QXP, the 2 lightsabers that Emma alluded to, it reduces that toxicity. All of the toxic, horrible chemicals that are produced during the print reaction and all of that constriction of space are alleviated, and the cells are much happier through the process. So it is difficult, and it's still quite difficult with the QXB, but it's significantly less difficult.
Okay, so you want to give the cells a little cuddle,
Yeah,
but not too much
exactly.
of a squeeze.
Yeah,
Right.
yeah.
Okay, that's my level of
Keep them
understanding.
happy.
Yeah.
Keep those cells happy. So I know that, I guess the technology has developed, and this Quantum X print is fairly new, but it's related to an EU-funded project that's just come to an end, Dave, that you were funded by. So can you tell us about that, the new chip project?
So that was a 2020 EU FET Horizon. So that was a project between Aston with Rain Parry and David Sard in the maths department, and some people at CNRS in Paris, some guys in Israel, some guys in Loughborough, and some guys in Barcelona, alongside a company called 3Brain. And we were using not this 3D printer, but a different version called photolithography to create these sorts of structures to grow cells of the brain in different compartments. And then we're trying to connect them in specific directions, like we see in the brain with the specific cortical loops that we have, to try and get directed connectivity. So the application of the project was going to be towards biological AI. So we could use what cells do to inform machine learning algorithms. So artificial neural networks that are going to be edited and use the information that we have from biological neural networks. So we would use stimulation parameters to try and connect areas together, whether that be chemically or electrically or optically with optogenetics. And We have just published one paper on that, and there's some more to come. The first paper came out in PNS Nexus the other day, and that was looking at how we connect these areas and mathematically modeling how these networks progress over time.
Amazing. So I guess an obvious question from that is, what does the Quantum X do that previous printer that you used?
Yeah, so the previous printer it used you etch a surface basically, and then pour a silicon-based compound called PDMS on top, and then use that as a mold. Whereas this printer will allow us to print in place. So if we want something in a specific place, like we grow cells on what's called microelectrode arrays, which can do electrophysiology of the cells. We can print that in the electrode array that we would just have to place it in there before. So it can be a more permanent way to get the cells to grow in specific locations and within a specific conformation that we already need. Also, this can allow much different materials. Before, it was basically just PDMS that we could use. Now with this, we can print with hard materials, soft materials, 3D materials, with a finer level of detail than the lithography could in the Z, rather than just in the X and Y. So it just allows a bit more flexibility and different studies based on material biochemistry and surface chemistry that we couldn't do in the past.
Amazing, thanks. So I want to move on to the specific science that you guys are doing with it now, starting with you, Emma. So Dave mentioned previously you're looking at the liver. So what are you proposing, or what will you be using the Quantum X to study?
So my liver research is based around the disease process that is fibrogenesis. So that's the chronic sort of scarring of the liver. And there's some key elements there that we find really difficult to model. And the sort of 2D things that Dave mentioned earlier, where we try and culture cells, even if we go to sort of simple 3D methods, we really struggle to recapitulate the things that we see in that chronic disease. And that's where this kind of level of scale could really help. So key factors that I'm interested in is maybe thinking about how we can use these specialized hydrogels to actually incorporate the extracellular matrix, so the scaffold that surrounds our cells, into a sort of biomaterial, and actually use liver-derived. So I have a PhD student now who's currently looking to do take the cells out of actual human liver tissue and use that matrix. And once we get more information about that, we can really inform that and how we print these scars and print mini scars using this kind of Quantum X technology. We can also do things like incorporate elements of stiffness. So as we age and as we head down the fibrotic routes, our tissues get stiffer, and we can really build that in quite a small area to go from quite soft tissue, like healthy liver, up into the more stiffer kind of regions of disease, and really model that process, and then build more elements in. So introduce some sort of flow. The liver has a unique blood supply, all of these things. So the short answer is absolutely loads of things. And it's always down to logistics of the people, the pounds, and the projects.
Sure, but it feels to me like a real bespoke system. You can really fine-tune the material that you want to put the cells in a specific environment, depending on whatever you want to study. It's amazing. Yeah.
Game changer.
Yes. So I guess, Dave, we'll come back to you next. So we've discussed the new chip project. So what are you doing in your research fellowship?
So what I want to do now is working on traumatic brain injury. And as Emma said, with the stiffnesses changes after injury, the extracellular matrix of the brain changes drastically. So I want to model that in 3D in a system that can only be made probably by this sort of printer, because we need it to be optically clear. We need it to be finely tuned with the properties of the polymer and of the cells and of what we grow them in and the area that they're growing in. So it's the sort of, this machine will allow, it's the only way, as far as I'm aware, to get these 3D models that have not yet previously been done. So we can use it for drug discovery or look for the mechanisms, what happens, and see what changes after the disease, and if we can after the injury, and if we can see if there's a way to prevent worsening of the injury, because we can't stop it happening because it's mostly done by like a car crash. But if we can prevent something getting worse, then this machine will allow us to look at that and study that. And I've got a PhD student starting this October who's going to be working on this and trying to make a blood-brain barrier model. So we can use, as Emma said, flow again. So we need flow through it, because everything in the body moves. We can't really do that in the normal cultures that we do, but this machine will allow us to add some microfluidics in there, so we get flow like a blood flow, or like the cerebrospinal fluid flow, so we can see what that changes, and we can look at how injury and that can model a drug target in the future.
Great. Yeah. Best of luck with the PhD project. Looking forward to following it. And then, Dom, I guess you have more of an overview of the broad range of things that are studied using the Quantum-X. So can you add a bit of another flavor to that?
Yeah, I guess you mentioned a really interesting term just then, Matt, is bespoke, and Dave was talking about flow. And there's two really interesting terms that sort of come together in the QXB for all the applications that you can sort of possibly get out of the machine. You're looking at things like Dave mentioned, sort of microfluidic devices. The beauty of the QXB is, again, if we go back to our jam analogy, our sculptor analogy, you're sculpting it a very small, then all of a sudden you can take it very, very big. So you can narrow the flow of liquid through chambers, thus creating pressure, forcing maybe proteins out of a solution. There's lots and lots of applications for that. You've created an artificial sort of capillary network almost. There's people with interest in drug discovery that are looking at various shapes, which shapes are best for controlled release of drugs. You know, some people, there's triangles, there's squares. Previously, as Dave alluded to earlier, you were casting and molding in quite logistically demanding processes to get all of your experimental conditions. On the QXB, you could have all of those experimental conditions on one chip. So you could have all of your drug release shapes in one place, and you're looking at the best one from the same condition. You know, you're talking about replacing animal models with very, very bespoke prints. The applications are very, very broad, and you know, I think there's going to be a lot of use from this system. I really do.
Great. That's the idea.
Yeah.
And then, so I asked Dave earlier, and he was talking about the kind of resolution that we can get down to the 100, 200 nanometers. So in terms of printing something that might model a bit of tissue, how big an area can be printed?
So on the QXB, every 3D printer has a sort of, they talk about X, Y, and Z. So that's horizontal, vertical, and then height. On the QXB, it's 50 millimeters X, 50 millimeters Y, and then 20 millimeters in the Z, which doesn't sound like a lot, but if you're thinking of 3D printing a tissue model, that's actually quite large. It's a big area that you can get, and there's a lot of cells and information that you can get from a print that sort of size.
Great. No, it certainly sounds like a good slab of material to do something with. Like you say, when you consider how many cells can be in there. Okay, so what I'm going to move on to next is kind of what we can look forward to from you guys in the future. So there's lots of cool research going on. I can clearly see that. And as a simple chemist in this terms, I find it fascinating. So what we want to do as AIM is, one, show off that we've got this instrument because we think it's amazing, but also advertise the fact that if you want to collaborate with us or engage with us in any way from academia or industry, there are mechanisms to do that. And Dom, do you want to give the email address?
Yeah, so it's QXB, the letters, _support at aston.ac.uk.
Yeah, and we'll put that on the screen, hopefully, and have it in the description of the podcast. So if this is a direct call to anyone who's watching, if you find this as fascinating as I do, and you have a purpose to kind of print some tissue models on this high resolution and this 50 millimeter by 50 millimeter by 20 millimeter scale, the maximum scales that we can operate at, get in touch. I'll repeat the email address again. So it's QXB_support at aston.ac.uk. And with that, it's time to wrap up. So thank you, Emma, Dave, and Dom, for great discussion. Didn't think we'd cover lightsabers and Star Wars, so that's amazing. And thank you for listening. I'm going to sign off by saying that Aston Institute for Membrane Excellence is supported by Research England. We're incredibly grateful for that, and also to the BBSRC for the alert funding that paid for the Quantum-X. So see you again next time.